Welcome everyone to Invest4Biotech. Invest4Biotech is Genopole's own demo day. We are very excited to have you all with us today. We know investors are coming from all around the world. Some are logging in very early, some late in the day. So thank you very much for taking some time in your busy day to attend this event with us. To start up Invest4Biotech, we have the pleasure to have first Olivier, who is the head of department at Genopole, and he will tell you a little bit more about Genopole. Olivier, the floor is yours.
Thank you, Hélène. Hi, everyone. I'm Olivier. I'm the Executive Director of Enterprise at Genopole. I just wanted first to express as well as gratitude to all participants, all of you, for this first edition of a fully virtual demo day. Which is both a bit scary because of the dependence on technology, but mainly exciting because it allows us to have you guys from all around the world. I just wanted to give you a very quick glance of what Genopole is. And in a nutshell, I would say that Genopole is the premier cluster in France dedicated to biosciences and biotechnologies, maybe in Europe as well. So you can see the physical aspects of that on the video in the background. So in essence, we're a campus where you can meet research labs from public and private research organizations, and also companies, a lot of different companies from startups, early stage startups, to corporate subsidiaries. But Genopole, and specifically our department, is mainly focused on the intangible aspects of this ecosystem. I like to say that we are both nurturers and connectors. We nurture companies, and especially we nurture entrepreneurs. 99% of our activities is dedicated to entrepreneurship. We nurture them through different programs, and I just like to emphasize two of them very quickly. The first is an incubation program dedicated to startups even before they are startups. We kind of track them down, track entrepreneurs down in the labs, in schools, in universities, in companies, and then we support them into bringing them into an entrepreneur attitude, an entrepreneur posture, and considering starting their own company. And the second program I'd like to highlight is a program called Gene.io, Gene.io in short, which is basically just an accelerator for biotech companies. And I'm not going to go deep into that because you all know the principle of an accelerator. But we also provide more transverse services to our companies, be it from internationalization programs. We've got a lot of partners around the world in order to provide market access strategies to different markets to our companies. We've got programs of connections with corporations, of course, which is quite critical in our industry. And finally, we provide a lot of access to funding and investment opportunities as well. And this demo day is kind of the highlight of that part of our program, which is led by Hélène. I hope you're going to have very productive, very fun, very interesting, but very productive most of all session with all the companies you're going to have here today, which have been groomed by us, which have been nurtured by us. As I was previously said, we are part of Genopole, which are the flagship of what Genopole can support in doing. COVID has been, of course, like for everybody else, has been a drag, but it has also provided maybe a lot of opportunities. A lot of companies are being formed. A lot of investors are being interested in what's doing in biotechnology. So I hope today's session will be the epitome of that for Genopole, and I hope you're going to have a very productive session. Thank you again. Thanks to all, and back to you, Hélène.
Thank you very much, Olivier. So like Olivier said, we're going to have a full pitch session. So how it's going to work is you're going to see a first startup pitch. And then during that time, if you have any question, feel free to send it through the chat box. After the pitch session, I will be the one reading the questions, and we will have around 5 minutes for Q&A. And then after that, if you are more interested to discuss with this company, of course, you have the partnering session. Some of you have already set up the account on the B2Match platforms, but you still have time. You still have time until tomorrow to set up your account and start partnering with our startups. So to begin the pitch session, we're going to have our first company. It's called Agenti, and it's very interesting because they're going to be using AI to help diagnose Alzheimer's. We can launch the first video.
Hi, I'm co-founder at Agenti, and we are on a mission to detect Alzheimer's early when it can be prevented. Today, 50 million people are suffering from Alzheimer's, and without prevention, it will be 150 million in 2050. And the bad news is, by the time we detect the disease, it's already too late. With the current diagnostic tools, we detect Alzheimer's during the late stages, when the symptoms are irreversible. And if we want to deliver meaningful treatment to the patient, we need to detect Alzheimer's from the asymptomatic stage with a high level of accuracy and a simple blood test. And that's exactly what we're doing at Agenti. We are a highly skilled team in neuroscience, artificial intelligence, and business. And you have research collaboration with 7 international memory centers in the US, Europe, and Australia. They give us access to plasma samples of people who have Alzheimer's right now, but have been sampled up to 15 years before. So Alzheimer's progresses over 3 decades. It starts with an asymptomatic stage where there is no symptoms at all. Then it goes through a prodromal stage with a subtle cognitive decline. And at the end, the dementia stage, where we diagnose today the majority of the patients. And for each one of these stages, the biomarkers are not going to be the same. So if we want to identify these biomarkers, we must have access to the brain and at the same time to the blood. And it's not possible in humans during the silent phase. To overcome this challenge, we use the first animal model of the silent phase of Alzheimer's, developed by my associate, Dr. Jérôme Brodeau. We sample the blood at each key stages. We analyze more than 2,400 plasma constituents. And by running machine learning algorithms, we identify and patented 105 biomarkers. After that, we validated these biomarkers on 232 retrospective plasma samples from France and Spain. So we had 2 groups, non-Alzheimer's, composed of healthy controls and other dementias excluding Alzheimer's, and Alzheimer's with patients in the asymptomatic, prodromal, and dementia stages. All of the patients have been followed up to the dementia stage. So how does it work? We take a plasma sample, we analyze 25 biomarkers, and by running a neural network, we achieved 98.8% of specificity and 98.1% of sensitivity during this first proof of concept. All of the computers are focused on brain biomarkers like A-beta or tau. And with this approach, they cannot detect Alzheimer's from the silent phase, and even during the prodromal stage, the level of accuracy is not so great. At Agenti, we're using peripheral biomarkers, so biomarkers that are not produced by the brain, but that are regulated by peripheral organs, like the liver. And we combine these multi-omics biomarkers with artificial intelligence to diagnose Alzheimer's from the asymptomatic stage with a high level of accuracy. We're planning to sell the test to the pharmaceutical industry to include patients in clinical trials as soon as mid-2022. And our objective is to become the gold standard for AD clinical trials in 2026. For the general population, as soon as we get the CE mark and the FDA approval by end of 2023, beginning of 2024, we will sell the test to the mind cognitive impairment population over 55 years old. And as soon as a large-scale prospective study is done, we will start selling the test to the population over 45 years old as a screening test. So we've already raised 720,000 euros, and today we are in a bridge round of 800,000 euros with 450,000 euros already committed. And we're going to use this bridge to do the industrialization of the test. So we're currently developing the targeted mass spectrometry protocol with Evotech. And we're going to train the algorithms on 750 plasma samples from the US, Europe, and Australia. And after that, we will do a final blind validation in 2022 on 500 plasma samples. But we're not only building the first blood test of Alzheimer's. Finally, we're building a platform to defeat Alzheimer's from the silent phase. And we can do that because we have access to the first animal model of the silent phase of Alzheimer's. and to thousands of human plasma samples with a clinical follow-up of more than 15 years. We are combining these two things with artificial intelligence to build a high-quality database, multi-omic database on Alzheimer's, and we use this database to develop 3 products. The first one, the blood diagnosis. The second one, a blood prognosis to estimate the conversion duration between the test and the dementia. And the last product, and maybe the most important one, will be to identify new therapeutic targets to treat Alzheimer's from the silent phase. So I hope you will join us on the journey to defeat the disease of the century. Thank you.
So now it's going to be the Q&A session, and we have a first question for you. So what is the market size for the AGNT test, please?
So we've got two markets. The first one is inclusion of patients in clinical trials. So it's the smallest market. It's around 125,000 tests per year. And the second market, the biggest one, is the general population. So the first one we are planning to target is the MCI population, so people with mild cognitive impairment due to Alzheimer's or another dementias. And this market, it's around 5.5 million of tests per year in the US and Europe, because we're planning to target first the US and in Europe, UK, France, and Germany.
Okay, great, thanks. So now we see how we can help diagnose Alzheimer's a little bit early during the silent phase. But what about the treatments? Do you have any prospects, any leads on curing or helping patients with Alzheimer's?
Yeah, so we are currently witnessing a major turning point in the fight against Alzheimer's from a therapeutic point of view. There are different molecules that are in the process of demonstrating their therapeutic effect, like aducanumab, for example. And the major shift is the fact that currently we include patients earlier than before. And we think that it will be the key here. If we are able to diagnose people before the reversible symptoms, we could apply the same molecules that we have tried to make work during the dementia phase, and they should slow down the disease from the silent phase. And another aspect is the fact that at AGNT, we have identified therapeutic targets, new therapeutic targets, peripheral ones, and by acting on it, we could slow down the disease from the silent phase as well. So in the next 5 to 10 years, there will be a drug on the market that will slow down the disease. We're sure of that.
Great. Thank you very much, Baptiste. We're very excited to have you at Genopole, and we're very happy to follow your development. And we hope that, yeah, it's going to be in the market as soon as possible to help everyone. Thank you, Baptiste. Have a good day and have a good partnering session. So next up, we saw that medication is really important, but beside medication, the formulation is also very important. And that's what we're going to see in the next pitch with Lamarck Biotech.
Hello everybody, this is Vaibhav Bhatia, CEO of Lamarck Biotech. We work with global vision to make medicines more accessible by creating advanced formulations of therapeutic proteins like insulin, vaccines and others. We are here to talk about a new pipeline that we are developing in age-related macular degeneration, which is a kind of a blindness which affects people above 60 years of age. So just to give you a taste, this is how a normal vision is. This is how you and I look at things. But people who go through AMD, this is how they look at things. And it's not small number. Around the globe, there are around 4.2 million people who are suffering this problem and are in the late stages. The numbers are very high for early stage and susceptible people. So is there a hope? Yes, there are therapeutic proteins which are being used to treat this for last 15-20 years. But the medicine has its own challenges. It's so difficult to deliver and maintain its efficacy. To give you an example here, the two most common drugs needs around 6 to 12 injections every year in the eye for the patient. It has its own side effects like cataract, and then even after going through all that pain, 25% patients do not respond to the treatment. There are these drugs, as I mentioned, and in the market, there are two breakthrough drugs called Lucentis and Eylea, which are mainly used for treating AMD. Good news, they are going off patent. And in addition, there are new drugs available with different institutes for out-licensing to target different pathways. But most of these, as I mentioned, are biotherapeutics, are proteins, and they are extremely large and extremely complex, which makes them delicate, sensitive, as well as it's difficult to create highly concentrated solutions of these pharmaceutical drugs. So what is the solution? Like a traditional formulation cannot have a highly concentrated drug, and it has low shelf life as well as low life in vivo, we create a formulation using our proprietary technology where these could be created, these formulations of proteins could be created at a very high concentration, and thus we create a kind of an intraocular depot of the concentrated proteins which are released at a very slow rate. What is the impact? It's only one injection compared to 12, and thus you improve the therapeutic value, the clinical value of the protein, which is already validated, and increase patient comfort. Are there any other companies in the domain? Yes, around a dozen companies are working to reduce the number of doses, but they fall in three categories. They either create an intraocular reservoir, they improve the stability and concentration, or they improve the efficacy by creating a co-formulation with an alternative molecule. And there are different companies working in different directions. Lamac falls at the center of all of them because our formulation do all of these three. So our business model is very simple. We talk to different stakeholders like labs or companies. We in-license a validated molecule. We use our technologies to create a formulation which is extra stable, and then we out-license it to a pharma company who has a market of its own in this field. We do risk mitigation by two major steps. One is we try to rope in an off-patent drug, like in this case I'm mentioning Lucentis or Xylia, and we start developing strategic alliance at a very early stage, so to have a potential buyer and a validated market. We are developing three lines. One is biosimilar, one is a new drug, and one is a co-formulation, because the two of them target different molecular pathways. In future, we plan to diversify in other areas like cancer also. So what's our development plan? We have extensive preclinical evidence in rodents for both the formulation as well as the drug substance. And we plan to file IND application as our first goal. And for that, we need to have in vivo toxicology studies, which we are planning to do in pigs. That's our first step. And followed by pilot scale batch production, and then followed by GMP batches, as well as filing of dossiers to go into the IND stage. The idea would be to make the drug fit for human consumption, validate it for clinical possibilities, and out-license it. What is the cost? So our investment plan is to raise 1.5 million in the seed stage, which we are raising 300K at the first feasibility stage, and then 1.2 million the later part, and which will be followed by 1.5 million for equity for strategic alliance based investments. We expect around 100 million of royalty as well as milestone payments coming from this line of work. We have a fascinating experienced team. I have been in AMD field in cancer biology for 7 years, and I've got 3 years of research business experience. I'm supported by Gavi as well as Luis, who have got 20 years of experience in protein chemistry, protein crystallization technology, which is central to our formulation technology. On our scientific advisory board, we are very fortunate to have people coming from different domains, mainly Narendra, who has got directorial experience in development of pharmaceuticals with Merck, Amgen, as well as Joel, who has been a serial entrepreneur and has developed many startup companies in France. So we here offer a promise of value-added biotherapeutics, and we have validated our market by creating alliances in different formulations. We offer strong intellectual property, validated intellectual property, and expected revenues, which are approximately 20x higher than the investment we are seeking for. We look forward to your question, and you can find us here online, as well as we are located in Genopole. So looking forward to talk to you more. Thank you.
Thank you, Vaibhav. So are you ready for questions? We have a first one coming in. So what will be your strategic positioning to capture the AMD market?
Thank you, Ellen. Strategically, we aim at two important things here. One is to use clinically validated molecule in parallel to preclinically validated molecule. This is to reduce the risk of drug discovery. And second is to be able to reach a strategic alliance early in our development. This is what we have done with a molecule in insulin, where we reached alliance with a big pharma company at a very early stage. The idea is to keep low risk as much as possible.
Okay, great. So you mentioned that you are looking to raise 1.5 million. Can you tell us a little bit more what you plan to do with this fund?
The first 300K would be used to define the critical quality attributes of the drug formulation. Mainly it will be used for animal PK/PD studies as well as immunogenicity studies, and also to create the process of production. Yes, they are the two main things, in addition to market validation that we are always doing. Yes.
Okay, great. All right, so I think that is all in terms of questions. Thank you very much, Vaibhav, and stay safe in India, okay?
Thank you, Ellen.
All right, talk to you later. Bye. All right, so next pitch, it's we're switching a little bit on the field, and then we're going into personalized medicine with cytogenetic tests. So next with cell environment.
It's a pleasure to introduce you my company, Cell Environment, dedicated to cytogenomic tests for personalized medicine. Cancer treatment is highly effective. However, some patients are at higher risk for resistance to the treatment, and once in remission for harmful toxicity. Personalized medicine based in individual genetic and epigenetic profile is a promise for clinicians and patients. To achieve this goal, reliable and robust genetic tests needs to be developed for the stratification of patients. Current clinical standards based on genomic approach are limited by three technical challenges. The first, the detection of repeated sequences. The second, the identification of complex rearrangement genes. And the third is the heterogeneity of the samples. All these limitations impair the patient stratification and their access to personalized medicine. Cell environment approach is combine new cytogenetic data to genomic data, and cytogenomic approach allows the detection of repeated sequences, the identification of complex rearrangement, and the detection of the heterogeneity of the samples for a better stratification of patients. Hence, you need the standardization and the automation of this new process in order to have a higher sensitivity in the certification of patients with rapid delivery of results and reduced cost. Currently, our high throughput cytogenomic approach has been published in 15 papers. The technology developed by Cell Environment is based in kit of staining, digital platform, and our service. And our first development concerned the standardization of our technology, allowing the production of 3 kits. The first kit dedicated to the telomeres, the second to the genotoxicity, and the third to the chromosomal aberration. Clinical validation of all these kits has been achieved. And the second development concerned the automation of the process via our digital platform for the analysis, ChromoDiag. 3 software packages are incorporated, and databases linked to ChromoDiag will allow the best stratification of patients. All this product has been patented. And moreover, our cell environment service provide various genetic tests. Our revenue models is based in the sale of the kit, the access to the GromoDiag, and our service. Currently, we have 13 clients. What about the market? The global market is the market of genetic tests. But you address another market, is the market of diagnostic tests in oncohematology with annual growth rate estimated to 40%. The position of the cell environment relative to the concurrence is based in three advances. Higher sensitivity related to the introduction of the repeated sequences, and short delivery related to the automation of the process, and the reduced price related to the miniaturization and automation of the process. Cell environment has been found by myself, and my management team is composed of Professor Patrice Card, oncohematologist, Dr. Eric Jean Didier, geneticien, Dr. Ili El-Malouf, computer scientist, and Wala Najar, assistant of research. In addition, cell environment is supported by scientific advisor with multiple background and different skills. Following all this technology development, the next steps are, first, the launch of our GromoDiag platform. The second is the consolidation of our cell forces. And the third is the development of two genetic tests. The first test dedicated to solid tumor, breast and ovarian, in collaboration with the French Society of Personalized Medicine. And the second test is the oncohematology test related to our expertise. Cell Environment has a worldwide cytogenomic community, and thank you very much for your support.
Thank you very much, Radia. So again, like I said at the beginning, if you have any questions, feel free to use the chat box and send us a few questions if you have. We're going to check if you have any question for Radia. So yes, what is the position of cell environment approach relative to single cell approach, Radia?
Yeah, the position of the cell environment technology compared to single cell technology, it's a very important question, because cell environment technology can be detected tumoral cells in tumoral cells and all other cells, and the interaction between all these the cells and the microenvironment of the cells. I think it's very it's more precise to the single cell. And you can perform many marker in our test many biomarker for our cells. Yeah, yeah.
Okay, okay, great. Thank you, Radia. We have another question for you. We saw in your pitch, we saw that this technology was you were using it for in the health tech. But is it possible to use your technology in another area?
Yeah, this technology can be used to another. Here I propose the cancer patients, but in genetic disease patients or in genetic population, and after the toxicology, after the irradiation or chemotherapy, etc. Yeah, you can apply this technology in many other areas.
That's really great. Thank you very much, Radia. It's good to know that that your technology can be applied in various fields in healthcare. So I think you already have a few meetings already planned, but I think you still have a few more slots. Is it right, Radia?
Yeah.
Yes.
Yeah.
All right. So feel free to log in and connect with Radia through the B2Match platforms, And yeah, Radia have a few slots available. Thank you very much, Radia.
Thank you, Hélène.
Talk to you later. Bye. All right, so we are staying in the oncology field with our next company, Sickle. I'm not going to tell you more about this. Uri is going to be introducing his company.
One important question when it comes to patients with cancer: what is the biggest fear? The treatment itself, the chemotherapy treatment. I'm Uri, CEO of Sickle. Happy to meet you. At Sickle, we developed a smart chemotherapy which is selective to tumors and safe for patients. Every day, 5,000 patients will be diagnostic around the world. Most of them will undergo chemotherapy, but we all know that chemotherapy has very strong adverse effects. Scientists have been working for the past 30 years trying to mix between efficient treatment and less adverse effect. Selectivity is key. It's exactly what we do at Sickle by developing a smart chemotherapy selective to tumors. We have a unique position into that specific market. We developed a double targeting of the tumor microenvironment. Let me show you how it works. We developed a molecular platform able to drive into the bloodstream active agents. That is enabling thanks to the liaison to the albumin, which is a well-known and characterized drug carrier. Let me show you how it works. We inject Sky1, that's its name, into the blood. It will link to the albumin and then becomes a bioconjugate, a macromolecule. And the macromolecule will go down to the tumor microenvironment thanks to two modifications induced by the tumor itself, solid tumor. The first one is a modification of permeability, which allows the bioconjugate to accumulate into the tumor microenvironment. The second modification is the overexpression of a specific enzyme. This specific enzyme, the beta-glucuronidase, present in the tumor microenvironment only, is responsible for the release of the active agent. Then the active agent gets into the cells and destroys the cancer cell. Again, only in the tumor microenvironment. We know that smart chemotherapy works. We have a very well tolerability. We know that even at high dose, we are efficient. We know that it's eliminated after a few days, avoiding long-term adverse effects. We looked at several cancers, strong cancer in the solid tumor arena. The pancreas cancer, it's one of the most difficult cancer because it's detected only late once the volume of the tumor is very big. As you can see here, we are able to decrease the size of the tumor. We are able even in some cases to cure the mice, whereas for the humans, it's almost impossible. We of course compared ourselves to the current treatment given to patients at hospital, standard of care. As you can see here on the blue curve, we are able to have no tumor whatsoever, whereas even with the standard of care, we still have a tumor with a big size. We look at the pancreas, but we also look at the triple-negative breast cancer, a very difficult cancer to treat too. Same way, fantastic results. All the invention and the data are covered by IP. We also look at the market, and thanks to the versatility of Sky1, we are able to cover several indications. We do have data for those indications. We compared ourselves, of course, to existing product or solutions such as the antibody drugs conjugate, and we are able to overcome those limitations induced by the ADC itself, but we see ourselves more as a toolbox, as a new element in the toolbox of the oncologist. We've been on our work for the past 3 years, closely working with our ecosystem. We raised 1.5 million over time. That allowed us to reach a situation where we have a complete and comprehensive package, which includes PDX, tox, pharmacokinetic, efficacy on several indications. I'm here today to raise 13 million in order to go for Sky1 into the regulatory phase and then into the clinics. At the end of the phase 2, we will be offering a license to a partner and then make return to our investor. Finally, we have also, and we need in order to bring that project to the end, we need a very strong team. I came myself from business development side, so I know how it works from the inside. Professor Papo, who's inventor, is won the Pierre Fabre Therapeutic Innovation for that specific invention. And at every single step of our development, we are surrounded by high-level technical and international experts, from the CMC down to the oncologist. It's a very active market. We are today ahead of the competition. So it's time now to join us.
Thank you very much, Uri. So now we have a question coming in that is wondering about your technology. So what makes Sickle technology so selective in targeting solid tumors, Uri?
Yeah, thank you for the question. I mean, the usual way of targeting cancer and solid cancer is really to look at a specific relationship between antibody and antigen. So if you don't have the antigen, you don't need the antibody, and for example, the ADC is useless somehow. What we have done is really to overcome that limitation by having a system which will recognize the tumor, be driven down to the tumor, and only at the tumor site be active. We don't need, for example, to get inside the cells, if we compare with ADC. We are very selective in terms of activating the relationship between the platform and the payload. The enzyme does the job, whereas, for example, other type of technology use a variation of pH. So all these elements together make several layers of safety and selectivity in terms of activation and recognition of the tumor microenvironment.
Okay, thank you very much, Uri. I think we have one last question, yes, coming in. So we saw that you did all your preclinical data and you have really pretty good results already. So what are your next steps?
Well, with the data we've generated in PDX, the next step for us will be to do the regulatory phase. And once we have done the regulatory phase, which is, as you know, an expensive phase because you need to produce the GMP GMP batches, and then to do the regulatory on the tox. Once you have all the data, you file the IND, and then you go to the clinics. That is the next step for us. We have all preliminary non-regulatory data that will be duplicated at the GMP level in order to go to the clinics. But all the steps and all the CMDO and all the partners are already on the starting block, waiting to get the money and to start tomorrow.
Great, thank you very much, Uri. You are starting your roadshow now, so really best of luck, and I'm sure you'll be able to reach your 13 millions of fundraising.
Thank you.
Thank you, bye, see you later. All right, so now we're changing a little bit in terms of field. We are now moving on with Innovem, who is tackling sickle cell disease.
Hello, my name is Marie Cambeau, and I am the CEO of Innovem. Innovem is a young startup which dedicates its research activities to sickle cell patients. The team is made of renowned scientists who bring their expertise in the red blood cell and its pathologies, and is complemented with an international scientific board and an infrastructure of established partnerships with academics and an essential network with industry leaders. Sickle cell disease is a very serious and chronic illness affecting around 4.5 million people worldwide. This disease is due to a mutation in the beta chain of the globin and leads to chemical and physical modification of red blood cells, causing their destruction. This is called hemolysis. Globally, life expectancy of patients is reduced by 30 years. In Africa, 50 to 80% of children are estimated to die before the age of 5. This is due to a lack of diagnosis and a lack of treatment availability. In Western countries, even though their lives are longer, patients face progressive organ damages that leads to a shortened lifespan because diagnostic tools are insufficient to provide an optimal medical care. At Innovem, our mission is to improve the care of sickle cell disease patients. By combining innovative biomarkers with artificial intelligence, Innovem will help the transition to a personalized medical care for each patient and will also help to develop effective treatments. Our biomarkers, single-cell fetal hemoglobin measure and a specific and direct method to measure hemolysis, are specifically adapted to the disease. Our product is called Sickle Bridge. This is a two-part product. The first part is made of the reagents that are necessary to perform the measure of the new biomarkers. And the second part is an algorithm of artificial intelligence hosted by a SaaS platform that will provide the interesting clinical scores. In comparison to standard biomarkers currently used, our product provides a better specificity, a better correlation to the disease, and is the only one to be useful as a prognosis factor. This will completely change the medical care of sickle cell patients. Sickle Bridge is a medical device for in vitro diagnosis, and in the market of sickle cell diagnostic, which is around 2 billion euros, our clients will be hospital centers, basically in Europe and in the United States, and in the market of sickle cell disease treatment, which is a huge and growing market anticipated to reach more than $5 billion by 2023, our clients will be pharmaceutical firms which developed new treatment to tackle sickle cell disease. In this market, the major players are those we are already in relationship with. So where we stand? We have already developed and validated the method to measure the new biomarkers, and we also provided a proof of concept for clinical correlation and for treatment monitoring. Now, to validate the clinical value of our product and to develop a SaaS platform according to regulatory authorities, we need a first fundraising of 500 kilo euros. We plan to access to European market by the end of 2023 and to US market around 1 year later. So if you want to be part of this meaningful project, we will be very happy if you.
Thank you, Marie. So now we are moving on to the Q&A. Are you ready, Marie?
Yes.
Okay, we can hear you.
Hello.
Hello. So yes, so can you tell us a little bit more about your industrial partners? Do you have anything going on already with some of them? Please let us know, Marie.
Yes, thank you, Hélène, for the question. Yes, we are already in relationship with some pharmaceutical industry because they're highly interested in the solution that we are developing. So actually, we already have an investigational research funding by an industrial partner which is Jazz Pharmaceutical. And we are also talking with a lot of others as Novartis, as Aruvent, also as Graphite Bio, which is a new stakeholder in the field of sickle cell disease. And all these pharmaceutical industry are interested in our developments because we are the only ones which can provide specific tools to assess the efficacy of treatments.
That's great. Thank you, Marie, for your answer. So you mentioned, yeah, there's already an interest in terms from some pharmaceutical company. because of your technology. Can you elaborate a little bit more about how your technology is so different from the current ones that they use?
Yes, of course. I mentioned during the pitch that we developed methods to measure specifically fetal hemoglobin and hemolysis toward the physiopathology of sickle cell disease. In fact, currently there is already some methods to measure fetal hemoglobin, but you have to know that this measure can only provide a mean in the level of HbF content in a red blood cell population. And this does not fit to the physiopathology of sickle cell disease, because fetal hemoglobin is a protective factor in this disease, and the fetal hemoglobin is not expressed homogeneously in all red blood cells. So if you want to know how many red blood cells, or the percentage of red blood cells that are protected in an organism, in a patient, you have to know exactly what level of HbF is in each red blood cell. So then you will have the percentage of red blood cells that are protected against the mutation. And then the other biomarkers that we developed is a new method to directly and specifically measure the direct product of red blood cell degradation. And now this is not the case. When you want to measure hemolysis, clinicians use biomarkers that are not direct. For example, bilirubin, which is not a direct product of red blood cell degradation, but can be also produced by other mechanisms. So there is false diagnosis of hemolysis when you use the current biomarkers, and you cannot precisely quantify hemolysis. With our technique, we can precisely diagnose hemolysis, which is a very important part in sickle cell disease.
Thank you very much, Marie, for your explanation. I think we have one last question. And you mentioned that sickle cell disease was really prominent in Africa. How are you going to address, how are your plans on how to address this in Africa?
So, yes, thank you, Hélène. Again, this is a huge problem, because most of patients are located in Africa, yes, but the problem in Africa is that the medical care of patients is not structured, meaning that there is not enough diagnostic tools. I mean, that patients are not diagnosed, basically. And after that point, there is a great lack of treatment availability. So now we cannot address African markets, because our tools are specifically developed when you have a structured medical care for sickle cell patients, meaning that this market is not mature now. So what we plan to do is first to participate to a lot of organizations to improve the care of sickle cell patients in all these countries. And then when this market will be as mature as possible, we will offer this solution to sickle cell disease patients in Africa.
Great, thank you very much, Marie. I think if you want to further discuss with Marie about Innovem and the future plans of development, you can still make a few appointments with her tomorrow. I think you still have a few slots, right, Marie?
Yes.
Okay.
That's right.
Great. So thank you very much, Marie. Talk to you later. Bye.
Thank you. Bye.
So we are kind of staying in Africa because our next startup is currently located in Egypt, and we are going to see how there's a new way to do bioproduction. So the next company is Proteina.
Hello, this is Mahmoud from Proteina. At Proteina, we are building the next generation biomanufacturing platform. So recombinant proteins now are everywhere. Flavors, vaccines, antibodies, biomaterials, and much more. They're a big market, more than $360 billion, growing at double digit annually. But there is a problem now. The current infrastructure for producing the protein of interest is not sufficient and not efficient. We are using now cell-based like bacteria, yeast, or other types of cells inside a seed tank that's being called a bioreactor to produce our protein of interest. Although it's validated, but there are some limitations. Whether it's complex, there are problems with scaling up the production, with the yield, and of course the cost. And we are solving that at Proteina. So we are replacing the bioreactor as a chassis that has cells with insect larvae as our own chassis. And we are solving a lot of problems here. So like in bioreactor, usually what happens in 1 liter bioreactor doesn't mean it will happen in 50 liter or 500 liter bioreactor. What happens in 1 larvae can happen in millions of larvae. Also, we don't have culture media here. We have the larvae has its hemolymph. We don't have to protect the larvae from any contamination. The larvae has its immune system. And we filed our PCT patent on that platform and also other provisional patents on other parts inside the platform itself. And we're proposing here a higher yield and lower cost. So we are proposing a 70x in the yield, comparing 1 gram of cells inside larvae to 1 gram of cells inside bioreactor, moving from synthetic conditions to natural conditions of the cells. Plus, a 50 to 80% reduction in the capital cost comparing an insect farm facility with a bioreactor facility. And the operation cost can be more than 90% reduced. How it works, we are working with a couple of insects. One of them is black soldier fly, very interesting insect to work with, not best, so it's rare, easy to rear. It can feed on any organic waste or organic byproduct. And it's growing very fast, more than 1 billion% in just 12 days from egg to a mature larvae. And this is our Insecta Pro platform. So we are starting with a protein of interest that we want to work with. Usually we are working with our partners on that part, developing a better vaccine or better antibody through our deep learning software. Then this goes to Proxy. Proxy is our deep learning software that helps us transform the insect and do all the optimization process for the production. Then we're delivering the gene with the egg, with the larvae, based on the protein of interest through our high throughput method. Then we are rearing the larvae in our farm, indoor, vertical farm, controlled environment. So we are controlling temperature, humidity, CO2 level, and much more. And then we do the downstream process to provide the protein of interest. This is our Proxy Engine. So in biology, usually we are starting with an assumption, which is a wrong assumption, and we want to reach the right one very fast. This is how we are trying to have a very fast cycle of design-based build-test learning, combining with deep learning model with automation to reach this cycle very fast. We have 2 main revenue models. One is PaaS model, production as a service model, producing proteins for others, or having our own product, or one with one of our partners. We are focusing on 4 markets: growth factors, animal health, enzymes, and human health. Our go-to-market is antibodies. So we now have a term sheet with a pharma company working with them on antibody for cancer. Also, we have ongoing projects. One is with Universals on having a new polio vaccine, VLP1, and also working with more than one having an edible shrimp vaccine. And we're also talking with other 9 partners who are having NDA delays or LOIs. So for partners, we are a member of Mesta in San Francisco, Genopole in France, Fraunhofer in Germany, also working with NC State University and Virginia Tech in US. We got our pre-seed round from 500 startups IndieBio, and Genesis for Sun 2. And now we are raising our seed round. We have a very diverse team. So we have Grow team working on the biology part, Wired working on the engineering part, Procoded working on the software part. We are now 17 team members between US, Egypt, and France. So we have team members with a very diverse backgrounds from aerospace engineering to AI, bioinformatics, bioprocess, biochemistry, and biotechnology. We also have a very concrete advisory board. Now we have 9 advisors in place with background in insect biology, insect biotechnology, downstream process, and innovation.
Okay.
And thank you.
All right. Thank you very much, Mahmoud. Mahmoud, I think you're here with us. Can you hear me?
Yes, I'm here.
Okay, great. So I'm checking in for the question. Mahmoud, can you tell us more about the downstream process, please?
Okay, so we have been tried in Proteina building the same downstream process being used for bioactors. So like now we only have one step that is very special for us for the insect platform, but all the rest of the downstream process is the same one is being used now for bioactor. So we can use the same infrastructure with any CDMO or CMO working with other bioactor technologies can be worked with Proteina too. So we don't have to have a new asset, especially for us.
Okay. Okay, great. Thank you, Mahmoud. There's another question coming in, and because you mentioned that your technology can be applied to a lot of different products, but which products are actually in your sweet spot?
So usually we prefer to work with complex proteins that are highly demanded. So like antibodies or vaccines that are complex proteins that being very hard to produce in bacteria or yeast. So we can produce them better on a scale, very cheap. So usually those are our sweet spot.
That's okay. That's great. And you can see right now in the current situation how important it is to be able to produce effectively and really rapidly vaccines. So
yes, true.
Yeah. So good luck with your fundraise, Mahmoud, and talk to you later.
Thank you, Helen.
Bye-bye.
Bye.
All right, so our next startup, we're switching field again, and we're going back into gene therapy, which is one of the main focus at Genopole. So we're really happy to have our next company with us. They're called FabMid.
Hello, I'm Clément Josset, co-founder of FabMid. FabMid is a biotech startup in the field of gene therapy. They are therapies that can cure severe disease with currently no treatments such as cancer, genetic diseases, etc. Thus, we are addressing a very large market, growing at around 30% to reach about $20 billion in 2026. So there are 2 types of gene therapy with each pros and cons. On the one hand, therapy based on viral vectors, and on the other, non-viral gene therapies. So let's start with viral vectors. They are quite efficient to deliver the gene in the patient cells and hence induce the therapeutic effects. However, they are very expensive to manufacture. To produce 1 batch of viral vectors, it costs between $500,000 and $1 million. So with 1 batch, you can cure only a dozen of patients. Raw materials are expensive, production yields are low, and you need to cultivate animal or human cells, which are fragile. Additionally, product scalability is also very complex. The way to produce enough doses for 100 treatments is the same as for 10 treatments repeated 10 times. This prevents cost reduction and a wider use of gene therapy. As for their therapeutic limitation, viral vectors carry a toxicity risk which further complicates their use on patients and manipulation. And finally, the limited size of the gene they can carry makes viral vectors less adapted for some complex diseases. Regarding non-viral vectors, they do not have these drawbacks. However, until today, none have been developed with an efficiency comparable with viral vectors. So at FabMed, we are developing a new vector combining the benefits of both viral and non-viral vector. So in practical terms, what is it? It consists of a circular DNA molecule binded to proteins. The DNA part contains the gene that needs to be delivered in the cells. And thanks to the protein part, it can easily target specific cells of the patients, easily penetrates in the cell and in its nucleus. As a result, it has an efficiency comparable to viral vectors. It carries no risk of genotoxicity, and it is customizable so that it can be fully adapted for each treatment. For example, you can use a certain set of proteins to develop therapy targeting kidney cells and another set of proteins for muscle cells. Regarding its production, it's based on much simpler manufacturing processes. For instance, you don't need to use any human or animal cells. So it is a scalable process and much cheaper, up to 10 times cheaper than viral vectors. Our clients, biotech and pharmaceutical companies, will be able to use our vector instead of viral vectors to develop their own gene therapy products. We will provide them with our vector for their R&D, and we will offer them licensing contracts and IP co-development partnerships. FabMed is the results of more than 2 years of research here at the Genopole. We have filed a patent at the European Patent Agency to protect our innovation, and an additional one is in preparation. We already have validation of interest by potential customers. We are currently a team of 2. My partner, André, has a long experience as a researcher. He has led a public genetics laboratory, and now he leads the scientific part of our projects. As for me, I have a background in consulting and finance, and I'm responsible for the business part. So in the next following years, we plan to carry on our R&D program to finalize our product and generate more IP. To this end, we need to expand our R&D team. We aim at signing our first contract with a client in 2023. To reach our goal, we are looking for investors that can support us during our development, and also for a fundraise of 1 million euros. So thank you for your attention, and hope to talk to you soon.
Thank you very much, Clément, for your pitch. So now we're ready for questions. I think there's one coming in. Yes, so your market is the gene therapy market, but it's a pretty large market. So which segments are you specifically targeting?
So our strategy is to focus on cell therapy or gene-modified cell therapy, and in particular the CAR T-cell market segment. When I explained that we aim to sign a first contract in 2023, it means for a CAR T-cell application. Why? Because within our market, it is a large segment with many potential customers. And besides, our technology is at the moment more adapted to ex vivo therapies, so cell therapies, than in vivo gene therapies. We will need more time, more research to have a vector perfectly fit for in vivo gene therapy. So to sum up, we will first focus on CAR T cells, then other cell therapies, and in the longer run, we will be able to address the in vivo gene therapy market or the whole gene therapy market.
Okay, good. That was very clear. Thank you, Clément. And yes, there's another question coming from an investor who'd like to know more details on what you're going to do with the money from your fundraising. Can you just give me a little bit more details on that? Thanks.
Sure. So we need to fund the continuation of our R&D program until we have finalized our first product in 2023. We plan to be a team of 8 people by the end of 2022 and 10 in 2023. So we will basically hire researchers and engineers. So the funds will be used to pay for their salaries. And we also need to rent a larger laboratory and buy quite expensive lab consumables for our experimentations. So all of this accounts for around 95% of the funds we need. So we want to raise €1 million and an additional 500,000 euros non-dilutive financing. And so this is the amount we are comfortable with to reach the goal we have set.
Clément, so you want to reach which point? I'm sorry. Can you detail more on that? Up to which point are you want to develop the kits?
I'm not sure to understand the question. So the fundraising we're planning now is to fund our R&D program for 2 years. And so our goal in 2 years is to have finalized these first products for CAR T-cell applications.
Okay. And then signing with a pharmaceutical company.
Signing a first contract and probably raising more money as Series A.
Okay. Thank you very much, Clément, for being with us. And yes, talk to you later. Bye. All right, so last but not least, we have our last company and last pitch. We're moving back into gene therapy, and this time we're going to see how AI can influence the development of gene therapy. Next with White Lab Genomics.
Hi, my name is David, and I'm going to present you with White Lab Genomics. We developed the next generation AI platform for genetic medicines discovery and development. So genetic medicines are life-saving, life-changing therapies. For example, this little baby, baby Emma, is diagnosed with a disease called SMA, and it's a life-threatening condition that could put her life at jeopardy. But thanks to treatment of genetic therapies, she will be saved, and now she's living and she's healthy. So how does it work? Well, genetic therapies are enabling the correction of a wrong genetic code here on the right side by the right genetic code to cure the patient from the disease. So that's pretty incredible, but the cost of those therapies is extremely high, and therapies like SMA treatment can cost up to $2 million to save the life of those children, as an example. Why is that? Is because development time of those therapies are very long, over 10 years, and they cost over $1 billion. So why is it so expensive and so complicated? Is because it's a scientific challenge. It requires to find the perfect correction and to include this perfect correction into the perfect vehicle to transport into the body. And it takes a long time because today the possibilities to find this perfect correction and this perfect vehicle are several dozens of million possibilities. The scientific, they have to navigate for many years between all those hypotheses, and they fail a lot. They use trial and error. At White Lab, we provide them with a tool to go faster through the different phases, hence they reduce the time and cost of the treatment. We do that with an unprecedented platform we've created that uses public data, biological information, multi-omics data, and proprietary data that we have developed ourselves. And it uses AI algorithms such as natural language processing, computational biology, machine learning, and federated learning. As an example, this is a flow for a researcher that wants to find a new treatment based on a specific disease, for example, here for Leber disease, for blindness. He has access to all this information through our platform in one click. This is another example of designing a new virus to transport gene therapy into the body by optimizing the design of the virus thanks to machine learning. Our technology is today a reality. We have already several real customer cases in different types of therapeutic areas whether it's cancer therapy, neurological disorder, or other type of diabetic disease. We are launching a platform that is using SaaS modules. We are launching 2 products, one called Power, one called Expert. Our market is the combination is at the crossroads right now of a very high growth market for cell and gene therapy, together with a very strong adoption of AI technologies in the field of life science. We have 2 different price points for our products, for Module 1 and Module 2, from $50,000 to $200,000, and for a total available market higher than $2 billion. Our revenue projections are to reach above 500,000 euros this year and go beyond the 1 million euro next year in revenue by selling our platform. And we have our target is to break even by the end of 2023. We already have strong tractions. We already have customers. We have already some sales, and we have pre-sales qualified and qualified leads. We are growing fast. We have signed partnership with large US companies, with large US academic centers. We're in discussion with the Korean consortium, and we already signed a partnership with key French players, just like INSERM and Genopole. And our team has grown from 2 to 12. This is our team. We are more than 10 people today, and it's a team composed of scientists, computational biologists, and computer scientists and computer developers. We are right now in our fundraising roadshow to fuel our growth. We are looking at a round of 2 million euros in equity to launch internationally our first module in the US and in Europe, and finalize the development of our second module by the end of 2022. Thank you for your attention.
Thank you, David. David, you're back with us for the Q&A, right?
I'm here. Thank you.
All right. So first question that came in, it's about the fact that there's a lot of AI companies that are also providing services for drug discovery companies. So what makes you different from all the others?
Yeah, so first of all, our platform is a SaaS software. So we will provide a B2B SaaS solution to our customers. And also, as mentioned, we don't specialize in small molecules. There are a lot of AI companies in the field of biochemistry. We are definitely in the field of larger molecules, DNA, RNA, so dedicated to gene editing, gene therapy, CAR T cells. So this is our scope. And in that field, it's a way less crowded still environment, and so we are one of the very innovative company in the field.
Great, thank you, David. I think we don't have any more questions, but I have one for you because I know that you are currently raising your fund. So could you tell us, how far are you? Is it still possible for new investors to come in? Do you have any amount already secured? Yeah, just tell us how far are you?
Yeah, we have already. We started about our roadshow about 6 weeks ago. We have a good traction with some investors. There's still definitely some room. We have already some people lined up, but we're happy to talk further with anyone who might be interested to join in.
Okay, great. Thank you. So feel free to make an appointment with David through our B2Match platforms. Thank you very much, David. I really like your hoodie. Very nice. I'll talk to you later. Bye, David.
Thanks. Bye.
All right, so we are drawing to the end of the first part of Invest4Biotech, which is the pitch session. So what remains is the partnering sessions that you can start actually right away after. Some of the companies have opened some slots for tonight. And if not, you have the whole day for tomorrow to do the partnering. All our startups are already online. I know a few of the investors have not opened their accounts, so feel free, you can still do it until tomorrow. Now that you have seen all the different companies, all the different startups, maybe now you are more ready to make appointment with a few of them. Last but not least, I'd like to thank everyone who's been involved in preparing this event. Of course, all the backup team at Genopole here at the studio, and all the startups who has been working with us in order to make this event as successful as we can. And now the only thing that is left is to wish all of you some fruitful discussions and hopefully some fundraising announcements soon. Thank you very much. Talk to you soon and at the next event. Bye.